I'd like to welcome everyone to the University of Nebraska Lincoln ARD Big Ideas seminar series. This series is we're focusing on advancements in plant identification and recognition systems, new technology, and what does that mean as far as our production systems, agriculture, natural systems that are managed or not managed in various degrees. Today, in our second speaker in the series is Dr. David Jacobs from the University of Maryland. He's in the Department of Computer Science, and he is working on plant recognition systems. He's been working on technologies for identifying not only plants, but other items as well. He is going to talk to us today about what he's been doing with computer vision systems, and in particular, you'll recognize the LeafSnap app, or an application that you can use to identify plants in real time. But I'm going to turn it over to Dr. Jacobs, and he's going to go ahead and talk about what he's been doing in this area.
Oh, thanks, Steve. So as Steve said, I'm going to talk about work I've been doing on species identification, and I'm particularly going to talk about work we've been doing on plant species identification, and I'll say a little bit maybe about animals. So I'm going to talk about this LeafSnap project first, and this has been to develop part of a long collaboration to develop mobile tools for plant species identification. And a lot of people have worked on this project. I want to particularly acknowledge Peter Bellumer, who's sort of led the effort on this project at Columbia University, and John Kress at the Smithsonian Institution. Peter and I are both computer scientists who work in computer vision, who've done a lot of work in face recognition before this. And so we sort of came to this project with that perspective of trying to do identification using visual cues. And John is a botanist at the Smithsonian, and so he's led the effort basically in helping shape the form of this app and also in collecting the visual data that we need, as I'll explain, for this application. And in addition to the three of us, most of the work has really been done by a large number of grad students at the University of Maryland and Columbia, and a lot of botanists at the Smithsonian Institution, by photographers and botanists at this nonprofit company called Finding Species. They've taken all of the really attractive photos that I'm going to show you, and a lot of volunteers who helped collect all of this data and who helped to build various components of this system. And so this is one of the most practical pieces of research I've done. Most work I do is a little bit more theoretical. And this has really been building a big system, and so it takes a lot of work to get something like this to actually work. So the way this project started was that Peter and I were sort of talking about things that we could do to use recognition in new domains, and maybe for some applications that were different from kind of face recognition, security applications. And we thought about using computer vision for plant species identification, and so we sort of just randomly got in touch with John Kress at the Smithsonian, and it turned out that they were really interested in a lot of the same issues, and in particular they were really interested in using, how all of these specimens that they had could be made into digital images and used in some productive way. And so we got a grant from the National Science Foundation, and we started working together on this. And the original impetus was to build tools for professional botanists. And they, at least, as I understand it, feel like there's a tremendous need for these kinds of tools. And it sort of went from there to something that might be useful for the more general public. But the first thing we did together was to just digitize the Smithsonian's collection of type specimens. So actually, how many people here are like from agriculture or botany? Okay, and how many people here are computer scientists? All right, okay, so computer science, okay, um, so I had no idea what a type specimen was when I started working on this, but I guess a type specimen is the original specimen that was used to identify a species. And the Smithsonian has this huge collection of almost 100,000 type specimens of plants. And so they'll take you back, and some of these were collected, by Theodore Roosevelt or something, or, hundreds of years ago. And the way botanists worked when we started this project, which was almost 10 years ago now, is that, some grad student in biology would go out in the field and collect some specimens, and then to understand those specimens and to analyze them, the specimens that they'd taken, they would want to look at the type specimens to help them identify the species of their specimens. So they would write to the Smithsonian, and the Smithsonian would send them a box of, maybe 100 specimens that they would borrow, these sort of 100-year-old, priceless specimens they would borrow for the next 4 years while they were trying to finish their thesis or something. So this is not the most efficient ways to use this data. And so the first thing, the thing that we knew we could do was just take high-resolution digital images of these and make them available through the internet. And that was the first thing that we did together. And although it's very straightforward from a technology point of view, I think it was a really valuable thing. And so moving on from that, the next thing we wanted to do was develop mobile tools to aid in identification. And we'd worked on this for a few years. And then when the iPhone came out, we decided to try to make an app that would showcase some of the research that we'd done on this problem. And so we created this app called LeafSnap. And the basic idea is that you can put a leaf on a plain background, take a picture of it, and use a search based on the shape of that leaf to try to identify the species. And so at the heart of this app is a lot of botanical data. And so for every species, we first have text descriptions of these species and some very beautiful high-resolution photos that were taken by this company Finding Species. And I think although this isn't technologically difficult, part of the value of this app comes from having these really useful, beautiful images that illustrate the leaves, the fruit, the flower, the bark, the petiole of these plants. And in addition to that, we also have images that are going to be characteristic of the kind of images that a person is going to take when they're trying to use the app. So the idea is that somebody's going to use this app by going into their backyard or going out in the woods, taking a leaf, putting it on a plain background, and taking a picture of it. And so in order to identify the species of those leaves, we have a training set. So for every species in the system, we have about 50 images of 50 different leaves showing the diversity of appearance that leaves from the species can have. And so I'm going to sort of begin by giving you a kind of tour of what this app looks like, and then I'll talk a little bit about some of the technology behind it. So this is the home screen for LeafSnap. And just sort of cycles through some different flowers, and then you can browse through different species of plants. So these are all different sorts of this is just covering right now the trees of the northeastern United States. And you can either browse looking at leaves or icons of the flowers or fruit. You can do text-based search. So you start typing in magnolia, and you can see the different types of magnolia that are in the app. And if you want, you can get more information about one of these species. So you can look at images of the leaf, the flowers, and you can zoom in on these. This is just to show you that these are really nice images with a lot of detail. Okay, and so we have these kinds of images for every species, along with textual descriptions. Now, if you want to use the app in this sort of more automatic mode, you can take a leaf, put it on a plain background, and take a picture of it. We segment the leaf from the background, and then show, based on the shape of the leaf, do a shape-based search. And just like a Google search, we show you the top matches, right? And so in this case, the top match for this is a Japanese maple. Second would be an Ohio buckeye. And you can see that there's some similarity to the shape of these top species, and if you want to, again, if you can look through more detailed information about these, the species that you're interested in order to help you identify the plant you're looking at. And so usually, when you're using the system, you might take a picture, you might look at the top few matches, you might decide that one or two of them look like they might be right, and maybe a couple of them look like they're completely wrong, and you're not exactly sure why you got that match. And then you maybe look more closely at those two that match well, looking at details that our algorithm doesn't capture. And this can help you. So the idea is to assist a person in doing an identification, not to build something that's fully automatic, which I think is still beyond our capability. And we also have some mapping tools so that you can identify, you can give identifications to images you've taken. You can you can keep a map of all the images that you've taken and look at where the different species of trees that you found occurred. Okay, so I think that's where Peter lives. So when we release this system, when we use the system, we take images that look sort of like this, a leaf on a plain background. When we release the system to general users, these are the kind of images that they upload, as well as some that are even sillier, right? So some of these are just people, yeah, actually these are all pretty serious. There's sometimes, you'll see pictures that people take of their friend's face or something. But there'll be some images like this that sort of fit the requirements of the system. And there'll be some images like this one where they're taking a picture of a whole plant, and while it would be great if we could identify that, we don't have the ability to do that now. And so the first thing we do is try to figure out which of these images we have a hope of identifying, and give the user some feedback if they haven't matched our specifications. And so we just train a classifier with many examples. So we took images that people had uploaded and labeled them as either valid or invalid just by hand. So we just went through and labeled a few thousand images as valid or invalid, and then trained a classifier to tell, is this leaf valid or not? And we find that this kind of classifier can work correctly about 98, 99% of the time. And so if the user takes a picture that isn't really, that doesn't meet our specifications more or less, we can just give them some feedback and say, we really want you to take a picture of a single leaf on a plain background, try again. And this helps both give people some feedback when the system isn't going to work as to why it's not going to work, and also help save us server resources as well, because running this classifier is very efficient. The next steps are a little bit more computationally intensive, and so we don't want to, we've got one server serving a lot of queries. We don't want to spend too much time on queries that we know we're not going to get right. And so once we pass that stage, this is some examples of some fairly typical kinds of images that we get. And you can see that in some sense, particularly if you do computer vision, you would say that this is a relatively easy problem, certainly compared to taking just a random picture and trying to separate out the different objects in it. But you can see that there's still some difficulties, right? We often have shadows associated with the leaves, and also sometimes leaves are somewhat damaged. And you can see there's a shadow here. by the stem. So the next step is to now separate this leaf from its background. And this is surprisingly difficult to get right all the time. So we do some, use some pretty standard techniques and we get good results, maybe 80–90% of the time, with still some challenges due to particularly shadows and highlights from very shiny leaves. And so this is the basic approach that we take to doing this segmentation. So first of all, there's a lot of work in computer vision on separating an object from its background. We call this segmentation, and this is sort of one of the classic problems in computer vision. We found that a lot of standard algorithms for this don't really work in our domain for a couple of reasons. One thing is that we need something very fast, and many of the best segmentation algorithms take seconds, maybe minutes, and we want something that'll run in about a second or less. But also, a lot of approaches to segmentation make the assumption that the object that you're separating from the background is a fairly compact thing. And with leaves, that's completely not true. We often have these very thin parts of the leaf. And so we can't really sort of use the overall shape and context with this compactness assumption to get good results. So instead, we use a segmentation approach that's just based on individual pixels. So for every pixel, we try to decide whether this is leaf or background. And we do this by first looking at these color images in a color space. So we can describe color using 3 parameters, RGB, or another common color space is hue, saturation, and value, where value is basically how light or dark it is. You know, if you made it into a grayscale image, would it be black or white? Hue is telling you sort of what's the sort of color it is. Is it red, blue, yellow? And saturation is telling you how much white is there in the color mixed in with the pure color. And it turns out, kind of surprisingly, that while value and saturation are really useful for segmentation, hue is almost completely useless. And this is, in some ways, a little counterintuitive because you would think, well, the leaf is green, the background isn't green, so the hue, the greenness, should be a very useful cue. But the problem is that when you take these pictures in the woods, there's usually a lot of reflected light that has a kind of greenish tinge to it. And so even though the paper looks white in these images, often it's got a slight greenish tinge to it. And so the saturation is really useful because that tells us the background is mostly white with a little bit of green. The foreground is almost pure green with a little bit of white maybe. And the hue is not very useful. So we represent each of these pixels as like a dot in this hue, in this saturation value space. And then we cluster all of these pixels into 2 groups. So if we take an image like this, we get dots that look sort of like this. And you can see that there's a big clump in the upper left, which corresponds to the background. And there's a region in the bottom, which is much more spread out, which corresponds to the foreground. And so we use the expectation maximization algorithm to cluster this into 2 groups. And this is an iterative algorithm where we sort of assign each pixel either to foreground or background with some probability. Sort of, you know, we say, this looks like it's mostly foreground. This looks like it's in between. This looks like it's mostly background. And then at each iteration, once we've made this assignment, we figure out where the cluster center is, and we do a new assignment based on that cluster center. And this kind of shows the beginning part of this where we have red for background and blue for foreground, and we're getting a segmentation that's not very clean. And then we iterate this a few times, and we get a segmentation like this, which in the original image space looks like that picture on the left, on your right. And so this works pretty well. with a few exceptions. So one problem with this approach is that sometimes, particularly in the case of pines, the leaf is very small, and so most of the pixels are background. And if we try to separate the pixels into two groups, because the foreground is so small, it's kind of negligible, and we just wind up separating the background into two separate regions. And so we have a scheme for sort of weighting the pixels that based on whether we think they belong, the likelihood that they seem to be from foreground or background, from leaf or background. And we do a weighted EM so that these pixels that are likely to belong to the leaf wind up having half the weight of the pixels, even though there are only a few of them. And so using that, we're able to get pretty good results on these kinds of very small leaves. So once we've done this segmentation, we then do some post-processing to sort of clean things up. So one thing that can happen in the initial segmentation is that if there are dark regions in the image, those can get lumped in with the leaf. So for example, sometimes when somebody takes a picture of a leaf on a piece of paper, when they take the picture is larger than the paper, and so some of the edge of the image is very dark. And because leaves are very dark, if there's a part of the background that's very dark, that gets grouped in with the leaf. And so we can just remove this using some simple heuristics. We just keep more or less the largest region that we find, and if we have a region that's mostly touching the boundary, we assume that that's background. And we also often get, because we're doing this segmentation based on individual pixels, often, there can be some pixels that are dark. Maybe somebody used a piece of paper that has a little dirt on it or something. And so if they're very small regions, we also assume that those are really part of the background. The next thing we do is remove the stem. And even though it seems like in principle stems should be very useful, at least knowing where the stem is in the leaf should be very useful in helping us to compare its shape to other leaves. In practice, when people pick leaves, sometimes the stem is very short, sometimes there's no stem, sometimes the stem is very long. And so it works out better to just remove any stems. And we do this, again, using some morphological operations. Basically, the stem of a leaf is typically a long, thin region that's sort of hanging off the edge of the leaf. So that if we remove that long, thin region, we're not changing the topology of the leaf. Actually, let me just illustrate that here. Right? So this is a long, thin region, but it's not the stem. Right? So if I remove this long, thin region, I'd be dividing the leaf into two parts. But if I remove this long, thin region here, even though it's not as long and thin, I'm not changing the number of parts I have in the foreground or the background. Okay, so this shows some examples of these different steps. So on the left column, we have an initial image. The second column shows the initial steps of the initial results of segmentation. The third column shows the results after we've cleaned them up a little bit and removed some isolated regions which were initially classified as leaf but are really part of the background. And then the fourth column shows after we've removed the stem. And so, for example, here we've got an image that somebody took with some of the background is really black. And so when we initially segment the leaf, that black part gets thrown in as part of the leaf, and we remove that. And then finally we remove the stem. Yeah?
Wondering about the shadow on the bottom.
Yeah. So this is an example where we didn't really get good results. Okay, so I didn't highlight that. But so down here you can see the bottom two of these are both examples where there's problem cases. So here the problem is due to that shadow, and you can see that the shape isn't really right down there. And in general, I think one of the areas that we can improve the most is doing better segmentation, and particularly with shadows. But it's tricky because they often look at least at the pixel level, very much like the leaf. They're just very dark, which the leaves also are, and they're very small regions often. So this shows another example where we have a problem, where we have a very shiny leaf, and so these highlights are almost white, and at a pixel level look like the background. And so you can see that this also winds up leaving a big hole in the leaf. So there's definitely some room for improvement here. Okay, so once we've done the segmentation, we're now going to compare this leaf that the users used as a query to all the leaves in our data set based on its shape and try to find species of trees that have similar shaped leaves. And we focus on the shape of the leaf because it seems like the most useful queue, single queue, and it's also in some ways, it's the easiest to extract. We can do this segmentation pretty well so we can determine the shapes of these leaves. There are other cues like the vein structure of the leaf that could also be very helpful but are harder to extract from images. And a variety of other cues that would be nice to incorporate. But right now we're just using the shape. And so this picture is just showing you 4 different species of trees and the shapes of the leaves associated with them. So these are very different shapes. And I'm particularly illustrating the fact that some species are very different at different scales. So if you look at the 2 leaves on the top row, at a coarse scale, those leaves look pretty much the same. The difference is the fine scale details, the serrations on the right. And on the other hand, at a coarse scale, these 2 leaves are very different. But at a fine scale, they're both very smooth. And so in comparing shape, we need to take account of both the overall sort of coarse shape of the leaf, but also at this fine sort of texture of whether leaves are serrated or not. And so we do that, actually, I'll just mention here, we do that using a multi-scale histogram of the curvature of the leaf. And so the idea is that if we look at the curvature of this leaf everywhere throughout the leaf, and the curvature of this leaf everywhere, we'd see that, well, at the coarse scale, the curvature here is pretty much, this is pretty convex, and the curvature doesn't vary too much. Whereas with this leaf, there's a lot of variation in curvature. At a coarse scale, the top 2 leaves are going to look like they have pretty much the same curvature, but at a fine scale, they'll have very different curvature, because at a fine scale, this is smooth and not curving so much, and the leaf on your right has much more curvature. So to compute curvature, computing curvature of shapes is a classic problem. And we use a method that's particularly robust to either noise or even to multimodal shapes, leaves that have multiple leaflets in them. And so the idea is that we take a small disk and just move this disk. Okay, so we take a disk like this, and we move this disk around the leaf. And at each location, we measure what fraction of the leaf is inside the disk. So what fraction of the disk is leaf and what fraction is background. And so here, for example, about half of the disk is foreground, half is background, and that's what you get when you have 0 curvature. When the curvature is positive, the leaf is convex, more of the background is going to be in the disk than foreground. And when we have a concave region, more of the disk is going to be foreground and less will be background. And while there are a million ways of computing curvature, if you have a perfect analytic description of your leaf, this method is particularly insensitive to any kind of noise, right? Because it's integrating a lot of information. And by varying the size of this disk, we can vary the scale at which we're computing the curvature. And so if we're using a really big disk, pretty much half of that disk is going to be foreground and half will be background. And so we can say that this is a pretty flat, pretty straight surface at a coarse scale, whereas at a finer scale, we'd pick up the fact that parts of that boundary are very convex and parts are very concave. So then we can, at every scale, we construct a histogram of all these different curvatures. This gives us a two-dimensional histogram, essentially, over scale and amount of curvature. And then we can compare two leaves by just comparing these histograms of curvature and saying that if the histograms of curvature are very similar, then the leaves are very similar. One of the advantages of this, so this kind of approach to identification has become very popular in computer vision, using other kinds of descriptions. So people try to recognize, for example, chairs or motorcycles using histograms of different sorts of features. And so it's not surprising that something like a histogram of curvature also works very well. This works well, and the other advantage of it is that it's very efficient. This curvature can be computed very quickly, and plus, we can compute this histogram of curvature for all the leaves in our data set ahead of time. We only need to compute it once for the query image, and then the comparison between the query image and all of the images in our data set is just a simple comparison of histograms, which takes no time. So we've done some controlled experiments to get some idea of how this can work in practice. We've used a data set for some of these experiments of 184 species of trees. Our current data set in LeafSnap is a little bigger than this. And so this gives you some idea of the diversity in shape of different leaves from different tree species. And for one particular species, this gives you an idea of how much variation there can be in the shapes. And so, but by collecting 50 images of each leaf ahead of time, we pretty well capture the variation in shape. Of course, some species have much less variation than this. Some pines, for example, every leaf just looks like a little needle. They all look the same. So when we do experiments to test the accuracy of this, we look at how often do we get the right species as the number one answer? How often do we get it among the top 2, top 3, top 4, top 5? And so this graph shows you that for that data set, we get the right species from a query leaf about 70% of the time. And we get the right answer among the top 5 species over 90% of the time. And we think top 5 is really, I mean, you might be willing to look at more than 5, but it's very good if we can get it in the top 5 or even the top 3. Now, one caveat here is that this test set is probably easier than the real life test set because these are real life images, but these were all images taken by us. And so they were all relatively good. And maybe better than the images that random users will upload. Okay, so we released this in 2011, and we got a really good response to it. And I think part of the reason for this is that, so there were a lot of newspaper stories about this, a lot of press stories about this. And I think part of it is that people are really fascinated by the idea of doing automatic recognition. And part of it is that there's definitely a lot of interest by enthusiasts, nature enthusiasts, who I think really genuinely find it very difficult to use traditional field guides and are really glad to have other kinds of tools to use in identification. So this was recently Scientific American listed this as one of the top 10 apps for your smartphone or tablet. And we've had about 1 million people download the app, which is completely free, so you can get it if you have an iPhone. People have taken about 1 million images. Although we only cover the northeastern United States, we've had people play around with it from other areas. And I have to say, we don't have a great way of telling who all these people are or how they're using it. But anecdotally, we know that, of course, some people just, are nature enthusiasts who've downloaded this. We also have people who've been using this for biodiversity studies. So just coincidentally, my son, who is an undergraduate at the University of Rochester, had some roommates who had a summer job doing some surveys of the forest in Rochester for the, I guess, the botany department there. And they were using LeafSnap to do this, not because of my son, but just because that's what they'd been told to use. And so we know some people have been using this for biodiversity studies, and we know that, but there was a picture there. We know some people have been using this in classrooms as well. So the, a middle school near me that one of the other faculty at the University of Maryland had kids at was using LeafSnap. We got some email from teachers in New Jersey who were using this, and I've seen a few websites of teachers using it. So one thing that we're interested in doing now is working with educators to use LeafSnap in schools. And actually, we've recently been working with Yale Weiner, who's at science educator, teaches science education at CUNY, City College in New York. And they've been trying to develop curricula for middle schools to use LeafSnap as part of the biology education. LeafSnap only covers the northeastern United States, and I think it would be much more useful if we could expand coverage to the continental United States. And there's not really any technical problems in doing this, but there's just the logistical problem of getting all the images that we need to cover. We have about 200 species of trees, and we need to cover about 900 to cover the continental US. And we're also have talked with people in some other countries about building versions of LeafSnap, and we're hoping that there'll be a version for the United Kingdom coming out next summer. We'd also love to develop tools for plant species identification in other regions like the tropics, where research botanists are really interested in identifying new species. So in the United States, you're probably not going to identify a new species of tree, but in places like the tropics, there's still a large number of plants who have yet to be identified, and tools for species identification might be able to assist in that. Tropical plants are much more challenging than trees in the northeastern United States. If you go to Costa Rica, in one region there might be 7,000 different species of plants, as compared to the 200 or 300 species of trees that you might find in one part of the U.S. And so it's probably what we're doing now would not work straight ahead in the tropics. We would need to use other cues like venation, and even then, this is a real research topic, but I think an important one. And another thing that we're interested in is species identification for things besides plants. And I'll just talk a little bit about some of the work we've been doing on that. So in the general problem we're interested in is species identification. And we've decided to use dog breed identification as a kind of model problem in addressing animals. And the reason why we picked dogs is that it's really easy to get a lot of data. So that's the first reason, right? So if you want to get pictures of Rottweilers, you can Google and get as many pictures as you want that people have uploaded onto Flickr of their Rottweilers. And in the plant domain, it took a lot of work to get all the images that we need, and so it's kind of nice to just be able to download everything really easily. The other reason to look at dogs is that they're a really challenging domain. As you can see, there's a big diversity of appearance in dog breeds, and dogs may have the most diverse appearance of any species of animal, or at least of mammal. So we view this as partly it's kind of fun, and partly it's a good test domain where we hope we can develop techniques that might be useful for doing species identification in insects and fish and birds and in other types of animals. So here's, so the main insight that we've had in comparing images of dogs and trying to identify species is that when comparing images like this, it's really important to compare corresponding locations in the dog, corresponding parts. And this is something we've realized in face recognition previously as well. So these are two dogs from two different species, and it's pretty hard to tell these apart, but the shapes of the nose are a little bit different, and the ear shapes, you can see, maybe that's a little easier to tell, are a little bit different. And so if we just try to compare some gestalt sense of the whole face, it's very hard to tell that these are different. It really helps if we can compare descriptions of localized features like the ears and the nose. Right, so it's a greater Swiss mountain dog and an Entelbuscher mountain dog. Yeah. So in doing this, we built up this data set of dog images that contains over 8,000 images, 133 different breeds of dogs. And this is giving you a sense of all the different breeds. And on the bottom, you can see a single breed and all the variation in appearance that we get within that breed. And then for each of these, someone by hand has localized the parts of that dog, where the nose is, where the eyes are, the tips of the ears, and a couple of other features related to the ears, where they attach to the base of the skull. So this is a lot of annotation. Luckily, it's become very easy to get this kind of annotated data. So there are these online services like Amazon's Mechanical Turk that allows you to set up jobs and ask somebody to label your data for a very small amount of money. And so you can get these kinds of labels for a few $100s. Okay, so this is just another example of how difficult this problem can be. So these are two different dogs that are the same breed, that are from two different breeds, right, but look very similar. And these are two images of dogs from the same breed. This problem is also difficult because we're not like flattening the dog and putting them on a plain white piece of paper. So these are just real images of dogs, and they could be in any kind of position. They can have very different appearances, and the position of their parts, even, and as I sort of hinted at before, we're focusing on facial features in identifying the dogs, but the position of these features can vary a lot. And I should say all these experiments are limited to images where we have a picture of the face of the dog, the head of the dog that shows both eyes. So we're not trying to identify the breed just from the body without the head. So here's a quick overview of how this works. We first build a dog face detector. And so we scan the image and find the dog face. Then we locate the eyes and the tip of the nose. And we use that to sort of normalize the position of the face. Then we build some description of the appearance of the dog in the region of the eyes and the tip of the nose. And we also infer the location of the ears. And so since we don't know what breed of dog this is, we try every possible breed. And for every breed, we make a different inference about where the ears would be if that was the right breed. And we build a description of the appearance of that location. So for the right breed, we're getting a description of where the ears are and what that looks like. And for the wrong breed, we're just getting some random description, which hopefully won't match anything. And then we do a query, this time instead of a shape-based query, a query based on these descriptions of the appearance of these ears and nose and mouth. And we, again, just as in LeafSnap, we show the user the top breeds. And this is just one way of presenting this breed. So all the breeds are labeled 15. This is breed number 15, which is a Basset Hound. And so on the bottom here, you can see the indexes for the different breeds that we're showing, and in this case, that green band shows that the first breed was correct. Okay, so I'll say just a little bit more about each of these steps. So to build a dog detector, we use techniques that are kind of related to human face detection. We take a little window and we slide it all around the image. So we consider this rectangle at every position, different orientations, and different scales. And for each possible position of that window, we have trained a classifier to say either this is a dog face or it's not a dog face. And the input to that classifier is a description of the intensity within different boxes around the eyes, mouth, and overall head, as well as a color histogram of the color inside this region. We use what are called SIF descriptors, which are a very popular way of describing local intensity regions. And we train a support vector machine to make a decision about whether this is a dog or not a dog. And so this is very similar in spirit to the way human face detectors and cameras work. It's a little bit heavier computationally. So the approaches that people take for human faces don't work that well for dogs, probably because the diversity of appearance in dogs is much greater than it is in people. And so we have to use a richer set of features and a more effective classifier to get good results. So once we found the face of the dog, we try to localize the eyes and the nose. And we do this using a kind of Bayesian inference, where we have some data, which is the result of a low-level detector that says, so for each feature, like the left eye, we train a detector similar to the detector we have for the whole face, which says, is the left eye here? Is the left eye not here? And those work pretty well, but they give a lot of false matches as well, and they don't capture the overall configuration of the face. So we might get a pretty good response saying the left eye is over here, and the right eye is over here. But we know that the left eye is always to the left of the right eye. We know that the nose should be underneath the eyes. And so we also want to capture this prior on the configuration of points that we expect for these point locations for dog features. And we use a non-parametric prior, so that is, we use thousands of examples of the configuration that these points can have. And we use sort of a big search to try to find the best way of aligning our prior with the bottom-up information from the detector. And so we can put this all together as a Bayesian inference where we say, our goal is to figure out the location of these parts given the detector response. And we can break this into a computation that separates out the location of each individual feature given the detector for that feature, combined with the difference in location between the inferred location of that part and what where the model says it would be. So it doesn't have to match our model exactly. It can match it with some amount of noise. And then we can view this as a big integration over all possible locations, over all possible models that we've seen before. And we evaluate this using a sort of stochastic sampling. And I just want to mention that this is exactly the same method we've used to find fiducial point locations of people in pictures of faces. And so these are some of the results that we get with exactly the same sort of system on some pictures downloaded off the internet. And so this is a system that's been trained to find the corners of the eyebrow, the top of the eyebrow, the corner of the nose, the bottom of the lip, and so on. And this produces really good results. And you can see, for example, it's even able to infer where the chin is in the picture in the upper right there, even though it's completely blocked. And this is because we're using this prior knowledge, different examples that we've already gotten of faces as our prior. And so the prior is able to infer location of points when there's no information from the bottom-up detector. So the detectors for the other features are sort of cluing us in as to where the overall configuration of points should be, and then we're using the prior to figure out where points are that are not actually visible in this image. And similarly, we're able to infer the location of the eyes when someone's wearing dark sunglasses. And this approach works well for a very wide range of head configurations, as well as different expressions, cases where the mouths are open or closed. Okay, so once we've got the location of the eyes and the nose, then we sort of cycle through every possible breed, and for each different breed, infer where the ears would be if this were the correct breed. So on the right there, we've got some example of a different breed that's been pre-labeled. So we know where the eyes and the nose are and the ears for that breed. We can line that model up with our query image and say, okay, if this were right, then we would know that the ears would be up top here. If this was the right breed, then we'd infer the location of the ears in a different place. So for every breed, we have a different set of ear locations as well as the eyes and nose location. And then we build, again, SIFT descriptors capturing the appearance of the image in locations around each of these features, around the ears, eyes, and nose. And we train a classifier for every breed saying, does this dog belong to this breed or not? So this shows some examples of results where on the left here, this is a query, and on the right is, in this case, for all three of these, we get the right answer as the top breed. And we're showing the next closest matches. And so you can see that in many cases, the next closest matches also look quite similar to the breed of the dog that we're looking at. Here's a couple of examples where we don't get the right answer. So on the top, the correct breed is second, not first. And this is probably just because these two breeds look very similar, and it's very hard to tell them apart. On the bottom, we're getting the correct breed as number 4. And if you look closely at these white dots here, you can see that we're not getting the eyes of this dog correctly. Right? So with sheepdogs, it's very hard to find their eyes. And so we're not getting the right answer here. At the same time, you can see that the top 3 that we're getting are kind of similar in appearance. Right? It's not totally mystifying as to why it picks those. So again, we can use this data we already have labeled to generate these performance curves to get a sense of how accurately this method works. So the top curve here is our method, and this shows that on this data, we're getting the right answer, the right breed first, about 68% of the time, and among the top 5, almost 90% of the time. And then these other curves correspond to other kind of baseline methods that we've tried applying to this. And the main difference between our approach and these baseline methods is this idea of localizing the parts of the dog and building descriptions around the parts of the dog, instead of building some general description of what the whole face looks like. And this is also just showing our method, different versions of our method. And in particular, if you do something just like our method, but without using this part localization, that's the green curve on the bottom. And so again, this shows that localizing the parts and building descriptions around the parts is really important. That's really the key thing in making this work better than previous approaches. Okay, so we've also used our LeafSnap infrastructure to make this into an app called DogSnap, which you can also download and play around with. One thing I've learned from playing with it is that it's very hard to get a good picture of a dog, particularly when it's not your dog and it keeps looking around for its owner. But if you can take a picture of a dog with its head visible, you can try this out. So you just, in this case, instead of putting the dog on a plain black background, we ask you to tap the nose of the dog. And partly this helps us to get more accurate feature detection, but actually mostly it allows us to crop and upload only part of the image to speed things up. And it looks, very much like LeafSnap. Right? It has the same browsing and the same kind of information. Okay, so thank you very much. This is what we've been doing on species identification. And as you can tell, there's a ton more to do. In particular, it would be really great to get other kinds of practical applications where we might be able to try, for example, identifying insects. Dogs are kind of fun, but there's a lot of very important applications to identifying insect species. Or we've been talking about weeds as another great potential application. And so we're looking forward to doing a lot more. Thank you.
Let's give David a hand. Thank you, David. It's very interesting. This is going to go beyond your normal hour. So for those of you who need to leave or want to leave, that's fine. But we are going to spend some time asking questions that you might have or that people have. So feel free to leave if you need to. But otherwise, does anyone have some questions for David? And if you do, let me don't talk until you use the mic so we can get for people that are watching online.
Before leaving, make sure you sign the colloquium attendance sheet.
Hi, is there any geographical information integrated with the LeafSnap?
So there isn't yet, because basically we just have one region. If we were to extend this to the entire US, it would be really important to use GPS to narrow down the set of possible matches and to get information on the region that every tree can appear. Because there's about 900 species of trees in the whole US, but in any one region there's no more than about 400.
How big is the Northeast region that's currently covered?
Basically from Washington up to Boston, say, and maybe through New York State, as far west as the end of New York State.
Thank you.
Does the scale of the leaf matter, or the size of the leaf, especially with that curvature method you were talking about? If you have a very big leaf versus a small leaf, it seems like that
Right.
curvature method would be sensitive to that.
Right. So what we do, we normalize all of the images so the leaf has a standard size before we do anything. And that throws away any information that there might be in, the size of the leaf might give you about the species. But since even within a species, depending on the time of year, the size can vary quite a bit, it's hard to make use of that size information very efficiently.
I was kind of wondering about if there's any work done using stereo or 3D images or possibly multiple images just to come up with some extra dimensional data.
Yeah, that's an interesting question. So I haven't seen work using 3D information for species identification. You would think that it might be, for leaves, of marginal value, because most leaves are flat, but it might help you a little bit with curved leaves. For dogs, maybe, or other kinds of animals, maybe more useful, and for whole plants, more useful. But it really limits the applicability of what you're doing, because everybody has a cell phone with a camera, but 3D sensors are still not that portable and widespread, but they're coming. So it seems like that's probably something that would be good to look at for the future.
I guess the reason why I ask is because a lot of noise can be detected using that kind of technique, and a lot of cell phones already have 3D picture taking capabilities. I I was also wondering, I had another question. I'll let somebody ask first. Go ahead.
No, go ahead.
To formulate it.
Okay.
I might have missed it, but in some cases you have a single leaf, whereas some cases you have like multiple leaves on a stem, and it's the conventional wisdom like leaves of three, let them be. So does it matter? I want to know how many times do you need the structure of the leaf topology versus just a single leaf?
Right. Yeah, so actually, everything I showed you is a single leaf. And so it doesn't look like it, but say here, for example, this is a single leaf and these are leaflets, which this is what botanists tell me. But what you said is true. I mean, having clusters of leaves would be helpful because sometimes the cluster really gives you a lot of information about species or whether the leaves are sort of opposite each other along a branch or whether they're alternating. And we're just not, that's one of a bunch of cues that we're not using yet.
You don't even need that.
Well, it might help, but we're not using it.
I'm going to ask one question. So it looks like one of the limitations is getting things to sit still or to be still or to not move to be able to identify them. If you're talking about dogs, insects, leaves, whatever, is there any way to address that, or do you see that technology developing in the future that we can take an image of something, record it, and then be able to identify it that way, or will that ever be possible, or what are your thoughts on that?
Well, so there's a lot of work, for example, on face recognition in video. And the simplest thing that people do is take a video and try to select images that are going to be most useful. And certainly, you could do that, right? You could take a video of a dog and try to extract out a single image where the eyes are both visible. But it seems like there should be a lot more information available than that, right? You should be able to integrate all this information. And there is some work on that with faces, so I would think that you could try something like that with other species as well. Yeah.
So it's coming.
Yeah.
David, how do you see the scalability when you go from 200 to, I was looking up, there are apparently 100,000 species of trees. How do you see, would you need to then go to these leaflets, go to more of a structure thing than just...
Yeah, so there's two scalability issues. One is, to cover the whole continental US, we'll have a lot more leaves, but that's not really a problem because within any one location, the number doesn't really increase. And so we can limit our search based on location. But when you go to regions where there's a lot more species, like the tropics, then I think this is not, this probably scales to a few hundred species. Once you have 1,000 to a few thousand species, you're going to have to use other cues, but it's challenging. Yeah.
So have you thought about things like hue that may not have been as critical now? Would that, do you think?
Yeah, my sense is that hue isn't, I'm not botanists can answer this better than me, but my sense is hue is probably not that useful. Vein structure is really useful. Other things like bark might be very useful. In my own experience in identifying trees, when the fruit and the flowers are there, they're a huge help. But the problem is that they're just not available year-round. So, of course, leaves aren't available all year-round either. But they're around more than the fruit and the flowers. And so, it's much easier when you can focus on one cue. And when you start talking about having to integrate 5 or 10 different cues, if we had the resources to do that, it would be fun to do. But it's probably any one additional cue is only going to help you a little bit more.
So is the computation right now done on the server side?
Right. So so this is, I didn't mention this, but this is a client server. So there's a server that all images are uploaded to. All processing is done on the server. And that's one of the reasons why we'd need it to be fast anyway if it was on the phone, but in order to handle a lot of queries at once, we need it to be fast.
For the leaves, we use basically the feature that we're interested in is the curvature of the leaf, and for the dogs also the position of eyes and other predetermined locations. I'm kind of interested in any work that you might have done in automatically recognizing features and then determining how effective that feature is in classifying, as opposed to wisely selecting predetermined options.
Yeah, that's a good question. Well, particularly for these kinds of features, which are really landmarks. Yeah, so we haven't really done that. I think it's an interesting question. I mean, for something like a face, it's probably not worth the effort to try to do it automatically because, I mean, particularly for all the people working on human faces, there's only one human face and you can do it. You can sort of figure out what the features are likely to be. But if you started dealing with insects, for example, it might not be at all obvious, at least to me, what the features are that are going to be useful. And it seems like that would be really interesting, but we haven't done that.
Yeah, I think hue saturation intensity can be worked, but basically the basis for hue saturation intensity, the mathematical basis is RGB, if you actually derive it. I don't know if that actually adds more overhead by doing that way. It all depends on how your initial images come in and how they're processed. Curious, you've had some feedback from users. How many images come in that are actually damaged leaves where insects have actually began to?
I couldn't tell you anything quantitative on that. I mean, my impression.
But they do come in.
My impression is that, yeah, you get some damaged leaves. It depends a lot on the time of the year. But certainly by September, you get a lot of damaged leaves.
Time of the year where perhaps maybe it would be more difficult to identify that way.
Yeah, and the way this is working, if the damage is in the interior of the leaf, it's not so much of a problem, but when it's on the boundary of the leaf, then it's a big problem.
Now, most of your work here, I guess this is all pretty much deciduous type trees.
Well, there's conifers.
Have you done some conifers? That's my question. Yeah.
Yeah.
So basically looking at a multiple leaflet type conifer.
Yeah, so the thing is that, I mean, the approach we're taking probably isn't that great for conifers because they tend to all look very similar. And the reason it's not killing us is because there aren't that many conifers. And so if the query is a conifer, we always get that it's a conifer. And so the right answer is usually in the top few because we don't have that many. If you had a lot of different species of conifers, you'd have to think about something else.
Two different questions. One, you're capturing the images when they're sending them in. Have you went back and did a verification to see what your accuracy rate is on user-supplied images from LeafSnap?
No, we don't really have any way of knowing with user-supplied images what the right answer is. So we have sort of anecdotal information that people. Well, here's my. I think with user-supplied images, it really depends a lot on who's taking the picture and how skilled they are. So some people love it and get really good results from it and can rely on it. Other people sometimes complain that it doesn't work very well. And I think the big difference is getting a feel for how to take the image so the system will work. And I think the first time you do it, it may not seem to work that well, but after you've used it a bit, it seems to work much better. And I feel like it works much better for me than most people because I wrote the segmentation software and I know exactly how to take pictures so it'll work. But I think that when it fails for user-supplied images, it's mostly because the segmentation is not working that well. And we do know that the segmentation on user-supplied images is failing a significant amount of time.
Like?
Somewhere between 25 to 50%.
The other thing I was curious about is probably not so much now that you're showing things are working and moving, but early on, you're obviously using techniques that aren't traditional botany methods for identification. Did you have much headbutting with your traditionalists as far as, you can do this, you can't do this, and how'd you handle that?
No, I mean, they were, the biggest problem we had with the botanists was that they had totally unrealistic expectations about how good we could do things, how well things would work. They just thought everything was going to work really great. But they never felt like we should do it the way the botanists do it. And I think, and there's a couple of interesting things there. One is that maybe what we're doing is capturing some information that are not in traditional field guides and the botanists don't traditionally use because they're hard to reduce to keys. And so maybe overall shape can be kind of difficult to describe in a key, but can be really useful. On the other hand, there's some things that are in traditional field guides used as some key features which we don't use because they're very hard to extract, but they're very easy for a person to judge. So, for example, whether the venation is open or closed in loops is pretty easy to see when you're looking at a leaf, but really hard for us to tell from, really impossible to tell from this kind of quality image.
That's a good question. We kind of addressed that last week in our seminar on the use of robotics in agriculture, and will that replace the farmer or the grower in these systems? And there's some issues with that, and realizing that developing these technologies is not to replace people, but to actually have them work within the system that's being developed. So I guess the question is, now that I've set you up, do you see this as replacing our traditional botanists?
Yeah, so that's a great point. So really, this is really meant to be a tool to help people in identification. And so the idea is that things like venation, a person can use, in conjunction with this. So I certainly don't see this as a replacement for a traditional botanist. My hope would be that it just can be a tool that, I guess I have the sense that in biodiversity studies, there are a lot of people who are not experts who are trying to identify species and find it very difficult. And even professional botanists, unless have a hard time identifying species that are not what they're expert in. So John Kress, who is an expert in ginger, and he was not the best one at identifying trees of the northeastern United States. There are people, unless you've really studied that flora, it's hard to be really accurate. It's not just like that. And so I think that it's helpful. The idea is that, though, that with a little bit of training, you sort of know what to look for. And if this can sort of hone you in on a small set of possibilities, it can be a lot more efficient than trying to use a field guide.
But also, I think the number of people who used to do this, excuse me. My understanding is that the number of people who used to do this, the number is shrinking rapidly.
Yeah.
So this is a way to help the fewer and fewer people who are doing this to do it in a systematic way.
Yeah, and
that's a help.
And the botanists talk about this, that there's a real dearth of expertise in species identification, and this isn't what academic departments focus on anymore. And I guess there's not a lot of money for it.
Yeah, it's on genetics.
Yeah.
Not as much in systematics.
Yeah.
That's the new technology. So, well, with that, I think I'll end. Does anyone have a question, or they just need to ask something? Okay, well, if not, I will end it. And thank you again, Dr. Jacobs, for your seminar.